Hello everyone. So today we are going to discuss about blue light sensing photoreceptors. So I have taught you about photoreceptors in the last video. So if you have not seen or if you have not watched that video, I recommend you to go through that video first, then come and watch this video. So I will give a brief introduction about the last video. We have discussed about photoreceptors. So you know that the sunlight not only act as an energy source, isn't it? They will act as a signal to mediate various photomorphogenic responses in plants. So in order to detect the signal or in order to detect the sunlight, plants have certain receptors called photoreceptors. So every photoreceptors they consist of protein part and a non-protein part except UVR rate. So this there are different types of photoreceptors. So you know that the sunlight light the wavelength of light is different. It can be red light, it can be blue light, green light. Okay, wavelength UVR light. The wavelength of light is also different. So this photoreceptors also are differ based on the ability to absorb specific wavelength. So based on that, photoreceptors are classified into phytochromes, phototropins, cryptochromes, seed loop and UV array. So phytochromes is a red light absorbing photoreceptors, red and far red, and they can absorb blue light and UVR also. And phototropin, cryptochromes, seed loop, they are blue light absorbing photoreceptors, UVR rate absorb UVB light. Okay, and they mediate various photomorphogenic responses. So now we are going to discuss about blue light photoreceptors. So mainly blue light photoreceptors are phototropins and cryptochromes. So they absorb blue light and they mediate blue light responses. Now what are these blue light responses? It include inhibition of stem elongation, then stimulation of chlorophyll and carotenoid synthesis, activation of gene expression, enhancement of respiration. So among mortal unicellular organisms, you know it's unicellular organisms such as certain algae and bacteria, this blue light mediate phototaxis. What is phototaxis? It is a movement of mortal unicellular organisms towards or away from light. Okay, so these are some of the responses that blue light mediates. Now, as I said earlier, this blue light photoreceptors include cryptochromes, phototropins, and seed loop. Okay, so mainly we can discuss about cryptochromes and phototropins. So, like phytochromes, cryptochromes also play a major regulatory role in plant photomorphogenesis. Phototropins, another blue light sensing photoreceptors, they are involved in chloroplast movement. It is important, it is there in the CSIR syllabus. C part question will be there. Stomatal opening, that is also important. CSIR syllabus, C part questions they are asking. And nuclear movement, solar trafficking, etc. Tracking, etc. Then seed loop family, they participate in control of circadian clocks. Circadian rhythm is important or it affects flowering processes in plants. Okay. So these are some of general introduction I am giving. Now we will discuss more about cryptochromes. So as I said earlier, cryptochromes are blue light for sensing photoreceptors. They mediate several blue light responses. Which are these blue light responses? It includes suppression of hypocortical elongation, promotion of cotyledon expansion, membrane depolarization, inhibition of petiole elongation, anthocyte production, circadian clock entrainment. These cryptochromes have sequence homology to photolyze. So you might have learned about DNA repair. Okay, so in prokaryotes, photolysis there is a one mechanism of DNA repair. Using sunlight, they repair their DNA. Okay. So these cryptochromes have sequence homology to photolase. That means it relate to some kind of evolution that we will discuss in another video. So in Arabidopsis, there are three cryptochrome genes. Actually, we are mainly conducting all the studies in the model organisms. Okay, we are explaining based on the model organisms. So Arabidopsis thaliana is a model organisms, and in CSIR syllabus also you have to study all the model organisms like Arabidopsis thaliana, sea urchins, sea elegans, Drosophila melanogaster. So many questions are coming with that. So in Arabidopsis, three cryptochromes genes are there: Cry1, Cry2 and Cry3. So Cry1 is the most stable one. They execute most of the cryptochrome functions. And Cry2 is unstable and Cry2 promote flowering. This is very important. Cry2 promote flowering. So you will learn about the detailed mechanism of flowering, how Cry2 promote flowering. That in actually it is explained in detail in another chapter that we will discuss in the coming videos. Then Cry3, they have photolase activity, and much thing is not known about Cry3. They are present in mitochondria and chloroplast. So Cry1 and Cry2, they are present in nucleus, and Cry3 present in mitochondria and photolase. No. So as I said that all the photoreceptors except UVA they contain a protein part and a non-protein part, chromophore. In cryptochromes, the non-protein part is a FAD or MTHF. FAD is flavin adenine dinucleotide and MTHF is methyl tetrahydrofolate. Coming to the protein part, it contains an N-terminal and C-terminal. So here we have a photolase domain, MTHF and FAD domain and CCT domain, cryptochrome C-terminal domain. So I will show you the textbook image. See in Arabidopsis, as I said earlier, there are three cryptochrome genes, Cry1, Cry2 and Cry3. So Cry1, cryptochrome 1 contain a photolase N-terminal domain. This is the C-terminal domain. So it is photolase domain, MTHF, FAD domain and CCT domain. But Cry3, they lack the C-terminal domain. They contain only photolase and MTHF, FAD domain. Cry2 promote flowering. So this is the non-protein but FAD and MTHF. This is the structure of FAD and MTHF. Actually what happens is that usually FAD is inactive. When they get blue light, what happens? This FAD is converted to FADH active form. When they get green light, it is converted to FADH minus inactive form. See the textbook sentence, Cry1 and Cry2, they are generally found in the nucleus and Cry3 is located in the chloroplast and mitochondria. So the function of Cry3 is unknown. It has been proposed that they have photolase activity. Now in this graph you can see that Cry1, this is the mutant, this is the wild type, this is the wild type. So if Cry1 is cryptochrome is overexpressed, you can see an increase in the androcyte accumulation. So here it is absorbent change of androcyte accumulation. So cryptochromes, I told that cryptochromes promote androcyte production. So if cryptochromes are overexpressed, you can see there is an increased production of anthocyanin. So in the B graph, as I told that cryptochromes will suppress hypocortical elongation. So if a mutant is overexpressed, you can see that the hypocotyl length is increasing. So what does it mean? So it means the cryptochromes actually they will inhibit or they will suppress the hypocotyl elongation. See, overexpression of cryptochrome 1 will inhibit the hypocotyl elongation as well as increase production of anthocyanin. So Cry2 play a major role in induction of flowering in Arabidopsis. Cry2. Now, you see the sentence, Cry2 play a major role in induction of flowering. Actually, how does cryptochrome 2 play or a major role in flowering? How they induce flowering? It is because this cryptochrome can bind directly to a transcription regulator factor, that is the CYP1. CYP1 is the transcription regulator factor. So when cryptochrome 2 binds to the CYP1, the CYP1 will bind to the flowering locus T, the promoter of flowering locus T, and when it binds to the promoter of flowering locus T, they promote the transcription of flowering locus and induce FT will induce flowering. Okay, so I have written in a note. I will show you the note. See how cryptochromes 2 promote flowering. So cryptochrome 2 have the ability to directly bind to the transcription factor CYP1. So CYP1 will bind to the promoter of flowering locus T. Flowering locus T is the transcription of flowering locus T is essential for flowering. So the binding of CYP1 to FT will promote transcription of FT locus genes and FT will induce flowering. So the detailed mechanism of flowering you will learn in another chapter. It is detail it is given that we will discuss in another video. So this now you just simply understand this thing. Next is see there is a co-action of two photo blue light for co-action of three photoreceptors: cryptochromes, phytochromes, and phototropins. So I told that Cry2 will promote flowering, isn't it? So for flowering, there is a co-action of cryptochromes, phytochromes, and phototropins. How? That is, so actually you know that Cry2 will promote flowering. This is the thing that you know. Cry2 promote flowering. Actually, the 5B, phytochrome B actually will inhibit flowering. Phytochrome B will inhibit flowering. So actually what cryptochromes do is that cryptochromes 2 will inhibit 5B. How? For the synthesis of 5B, there needs a transcription gene of ELF3. ELF3 is early flowering 3. So ELF3 is needed for the production of 5B. So what Cry2 will do is that Cry2 will inhibit ELF3. So if ELF3 is inhibited, there will be no 5B. So it will induce flowering. Similarly, but phytochrome A will promote flowering. Phytochrome B will inhibit flowering and phytochrome A will promote flowering. How is that? Phytochrome will stabilize a constant RNA. So phytochrome A will stabilize a constant Actually, it is important for the transcription of flowering locus T. So I have told in the earlier slide that SIB1 cryptograms do have the ability to bind to the transcription factor SIB1. Together with SIB1 and Cry2 will bind to the promoter of flowering locus T and induce the transcription of flowering locus. This will induce flowering. Okay, so phytochrome A will promote flowering and phytochrome B will inhibit flowering. How is that? Phytochrome B will degrade constant mRNA. This constant mRNA is degraded. So phytochrome B will inhibit flowering. Actually, cryptochrome 2 will inhibit phytochrome B. How? They will inhibit ELF3, which is needed for the synthesis of phytochrome B. That is the concept. So this is very important. CPAR questions they are asking based on this thing. See, this is the textbook sentence. So this text is the Taiz-Zeiger 6th edition plant physiology textbook. Phytochrome interact with cryptochrome to regulate flowering. So cryptochrome 2 probably promote flowering in blue light. Actually, in this textbook, they have written in a research style. Okay. So actually, that is the standard way of writing the textbook. It is not that much student friendly, I don't think. But you need the help of someone else to understand the text. So cryptochrome 2 will promote flowering in blue light and they will repress phytochrome B. How is that? Cryptochrome will inhibit phytochrome B by suppressing the activity of early flowering ELF3. Actually ELF3 is needed for phytochrome B. So if ELF3 is inhibited, what happens? Phytochrome B will not be produced, so it induces flowering. See this graph, you can see this is in a y-axis relative growth rate and time. So first if you initially the growth rate was very high. So when you induce a blue light, first actually uh, phototropins will act. That is another photoreceptors that we will discuss in the later section. Then only cryptochromes will act. So phototropins and cryptochromes, they are the two blue light mediated photoreceptors. So when the blue light is given, first phototropins will act, then only after sometimes cryptochromes will act. The light input appears to modulate the genes ELF3 and time for copy. Actually, tick tock, time for copy and talk, they are the proteins involved in circadian rhythm. So actually, uh, for the promotion of for there are many factors involved in uh, the flowering. For flowering, there are many factors are involved. So it includes day length perception, it includes circadian rhythm, hormones are included, cryptochromes 2 is included. So circadian rhythm, so two proteins are involved in circadian rhythm, that is tick-tock, time for copy and talk. And Jai you will be learning, actually this is explained in the coming chapter only. So everything is given together, so you will have some doubts initially when you read the textbook. Don't worry, that we will explain in the coming chapters. Now next is how next we are going to discuss how Cry1 promote photomorphogenesis. I told that cryptochromes 1 mediate most of the or execute most of the photomorphogenesis. So how they involved in or how they promote photomorphogenesis. So in this picture you can see this is the light, that is during the day, during the light time. This is during the dark, a moon is given. So this is the center, that is the first thing that you have to understand. Okay, so usually SPA1 and COP1, they form a complex in the dark and they will add ubiquitin tag to hyphae. So what is hyphae? So hyphae is a transcription factor for which is required for photomorphogenesis. So to execute photomorphogenesis for blue light mediated response, actually hyphae is needed. Hyphae will bind to the DNA and they will execute certain functions. So during the dark, that means you don't have blue light, or we can say in the absence of light or in the absence of blue light. Actually, we don't want this hyphae, isn't it? So SPA1 and COP1 will bind together and they will bind to the hyphae and they will add ubiquitin tag to hyphae. So this hyphae is degraded by 20S proteasome degradation. Understand? So actually the cryptochromes, I told this cryptochrome Cry1 has N-terminal and a CCT domain. So no one is there to absorb because we don't have light in the dark. So they cannot phosphorylate C terminal. So this is inactive. But what happens in the light is that when light or when the sunlight comes, there is blue light. So the light will be sensed by the N-terminal domain. Okay, so this Cry1 will be active and there will be some conformation change and C terminal will be phosphorylated. And to this phosphorylated C terminal domain, SPA1 and COP1 will bind. Sequestration of SPA1, COP1 to CCT domain will make free hyphae genes. So hyphae, no one is there to add ubiquitin tag to hyphae. So hyphae gene will be free. So it lead to the transcription of photomorphogenesis. That is a blue light mediated genesis. So hyphae will transcribe photomorphogenesis and will express proteins and it will mediate certain blue light mediated responses. Now another case is that what happens if CCT is overexpressed or mutant in dark or light? So this is the CCT. This is the CCT. So what happens if CCT is overexpressed or it is mutant? So that means it is always in a phosphorylated condition. So always this SPA1 and COP1 will bind to the C terminal and hyphae will always free and always this transcription photomorphogenesis occur and always this blue light mediated response will occur. Whether it is dark or light doesn't matter, always this blue light mediated response will occur. Actually, this was asked in one CSIR question paper few years back. One C-part question. What happened? This is the same way. What happened when CCT is overexpressed or mutant? So what happened? Always CCT will get phosphorylated. COP1 and SPA1 will bind. Sequestration with COP1 and SPA1 to the C terminal always activate or promote the transcription of hyphae genes. So that's all about cryptochromes. We have studied about cryptochromes in this video. We have studied about the various blue light responses, photoreceptors. Then we have studied detail about cryptochromes, his blue light responses, what are the blue light responses, and graphically how they are represented. We have studied about Cry1, Cry2, Cry3, three forms of cryptochrome genes in Arabidopsis. And we studied about the protein part and non-protein part, isn't it? Then what happened when the chromophore, FAD and MTHF, what happened when blue light, how it is converted to active form. Then we saw these two graphs. Then CRY2 proteins, how CRY2 proteins promote flowering. What is the mechanism? Then we study the model of cryptochrome interacting of carbon and how they regulate photomorphogenesis. This is actually blue light, okay. So these are the things that we have learned. And next is the phototropins, another blue light mediated photoreceptors. Actually here you will learn study about this chloroplast movement. It is very important, especially C-part questions they are asking. Chloroplast movement. This is a chloroplast movement. Then you will learn about stomatal opening. This is again a blue light mediated response. This is also very important for C-part questions, CSIR. So you will learn the detailed mechanism in the next video. So thank you for watching my video. So if you like my video, please like my video and share and subscribe my video. So if you subscribe only, you will get the coming videos. So thank you. So don't stop preparations. Keep on practicing. Read good books. Definitely you all will score CSIR. Thank you.